Good afternoon, everyone. Today is my pleasure to introduce you Dr. Enrique Quesada-Moraga. Enrique is full professor of agricultural entomology in the Department of Agronomy at the University of Córdoba. Dr. Quesada is very well known internationally for their full record of scientific publications, focusing mainly in the control of insect pests. He's particularly engaged in BEXYL project in Work Package 5, focusing on the development of biological methods for managing population of Xylella fastidiosa vectors. Apart from this prolific scientific career, I'm happy to say that Enrique and myself, we know each other for many, many years since we were classmates at the University of Córdoba. And we had followed very parallel careers. I'm focusing on plant pathology and he has been focusing on entomology. So Enrique, I'm really happy that you can participate in the Xylella seminar series. His talk will focus on developing strategic options for managing Xylella fastidiosa vectors with the use of entomopathogenic ascomycetes. So Enrique, normally we have 40-45 minutes, and then we have normally many questions. So you are free to speak when you want. The floor is yours.
Thank you so much, Blanca, for giving me the opportunity for this seminar. It's my first time within the frame of the Xylella seminars. I would like to thank also to all the people assisting. It is in Spain 6 o'clock, but I understand that probably for several of the people joining the seminar is probably it's not the best time. First of all, I would like to thank, of course, the BEXYL project, and within the frame of the Work Package 5, also to Professor Alberto Ferreres and Aranzazu Moreno, and of course to the rest of the people from the Work Package 5, Domenico, Paula, Ensa, and of course to my team, because some of the results I'm going to present, of course, have been possible with the collaboration of in the frame of a multidisciplinary work with all these people. Probably also discussing the Xylella problem in a Xylella focusing seminar may be to somewhat redundant. Let us really revisit the issue to better understand the strategies for applying entomopathogenic fungi. As you know, probably Xylella fastidiosa is a gram-negative xylem-limited bacterium. It's an obligated endophyte that usually may live in two environments: the plant host xylem cells, and of course the most part of the insect vectors, with many host plants. After the issue of the Puglia problem in 2013, causing the olive white decline syndrome, presently is a especially observed pathogen in the European Union, and of course is included in the priority list of the most threatening plant pests. Xylella fastidiosa is transmitted to plant by xylem feeding insect, belonging most of them to the order Demiptera, suborder Cicadomorpha, and the superfamily Cercopoidea, including the spittle-bugs and froghopers and Cicadidea, the cicadas, and also to the family Cicadelidae, particularly the subfamily Cicadelinae, in which they are included the sharp softers. Probably the two most important vectors to now describe it are Philenus spumarius and Neophilenus campestris. In Europe, I have to say that in Spain, Philenus spumarius is most frequently found, let's say, in the north of the country, whereas Neophilenus campestris is the most important species in southern Spain, particularly in the region of Andalusia. Probably the need for a cold temperature for development of diapause is a key factor explaining why Philenosis spumarius is less frequently found in southern Spain. And with this insect feeding the xylem sap or infected plant, the Xylella fastidiosa cells bind to the cuticle of the vector foregut, Presivarium and Decivarium, multiply inside the vector foregut, forming a persistent biofilm. And then, when feeding in a healthy plant, a biofilm, this biofilm in the xylem vessel walls block the transportation of the xylem sap, and this is clearly a dysfunction of the vascular system, and they have the problem. Here you have the Philenus life cycle, mainly in southern Spain, related to the relationship between the covers, either artificial or natural cover crops, and the olive tree phenology. This is a key point to understanding the strategic options to manage the problem of the vectoring of the Xylella fastidiosa by this homopteran. Obviously, this is a key moment in which the vector adults left the cover, the natural cover or artificial cover, and go to the pre-reproductive adults in summertime, go to the olive, in which the olive is producing new branch, and this may feed. And this is a key problem because in this moment is when usually the transmission occurs. Afterwards, in the autumn, given that the maturation of the branch are not very good for the vector, they come back again to the covers for reproducing and for laying. And of course, this moment in the springtime is a key point for the transmission of the bacterium. Regarding entomopathogenic fungi, we have no time for deeping of this microbial control agent, but there are three main aspects I would like to highlight because they are key to understand how we can manage these microorganisms for Xylella fastidiosa vector control. The first one is the mode of action of entomopathogenic, particularly the ascomycetes. And this is regarding why they are so important for the possible management of these vectors. We are dealing with pear-sucking insects, so the fact that the entomopathogenic fungi are contact action microorganisms is a key point. And this is why probably they are among the only strategy regarding microbial control for the control of homopterans. The infective propagules, the conidia, adhere to the cuticle. Here you see the cuticle of the insect, and they germinate. They may form an appressorium. Sometimes it's not the case, but in any case, they penetrate through the integument, through the cuticle, reaching the hemocell and infecting all the tissues. Here you see infection penetration points. Obviously, the fungus has to win the battle, the defensive battle, either humoral and cellular of the insect. And finally, usually you have usually if the strain is infective, you have the typical fungal outgrowth on the cadaver because the fungus again make the way back, penetrating the cuticle to form new spores, new conidia, for dispersion and for auto-dissemination in the environment. This is a Philenosis spumarius, by the way, infected by Metorrhizium and Isoplia. Here you have the stilette that is also infected. There are a market of entomopathogenic fungi. In this situation, when they are commercially available, we deal usually with myco-insecticides. But during the during this cycle, there have been many research in which my team has been an outstanding thing in the world regarding the role of entomopathogenic fungi presently as multi-purpose microorganisms, because they have been found as epiphytes in the plant, including the olive tree, epiphytes in the olive tree and the covers of the olive crops, as endophytes, and are rhizosphere-competent microorganisms. And this is why the strategic options for managing Xylella fastidiosa vector with entomopathogenic fungi are diverse. First of all, we have the most typical one, we could say that is the direct pest control. With direct pest control, I mean using the fungal conidia, the conidia suspension, the microinsecticide for spraying, directly spraying either the nines or the adult of the vectors. It could be that this is the typical mode of action in which the only objective we have is to make the spore reaching the epicuticle of the target Xylella fastidiosa vector. But considering the other roles, we have some other option because we can try to reach the vector not directly with the fungal suspension, but through the plant. We can try to make the fungus colonizing the plant, or either priming the plant and having an effect on the vector through the plant. This is either by fungal sprays onto the cover crops or fungal spray on the soil, or even by producing plant colonized by these fungi, either by seed dressing, by multiplication, the typical multiplication schedule, the nebulization in the olive crop. So remember these two important because my speech had been divided in how to proceed accordingly to these two different options. I mean, targeting the vector directly with the fungal suspension, with the fungal product, or targeting the nine for the adult through the cover crop or through the plant, through the main crop that is the olive. Consequently, the strategic option using entomopathogenic fungi for Xylella fastidiosa control, that by the way, they are all of them being explored within the frame of the Xylella of the BEXYL project, mainly in the Work Package 5, are the direct and indirect effects of fungal conidia sprays targeting vector nymphs and adults. And you have to consider that when we target the nymphs and the adults, or the adults, either or both of them with the fungal suspension, we can have either lethal and sublethal effect on the vector population, but also, quite important, we can have a possible interference on the vector Xylella fastidiosa transmission. Both aspects are very important to reduce the population of vectors, but even if it is possible, it will be even better to alter or to impair the transmission of the Xylella fastidiosa by the infected insects. In addition, we can target the vector or nymphs or adults through the plant, I mean, through the key plants in the olive crop ecosystem, I mean, the cover crops, either natural or artificial, or by the main crop, I mean, by the olive plant. And in this case, also, we can have direct lethal and sublethal effects on the vector population and the possible interference on the vector Xylella fastidiosa transmission. Then we are going to try to illustrate very briefly the main result we are having, because some of the experiments are ongoing, mainly those regarding the crop plant, the olive tree, which are the main result we are having regarding all these possibilities. The main results regarding the target of the nines of adults directly with the fungal suspensions of the entomopathogenic fungi, the result regarding the lethal, sublethal, and possible interference with the transmission when we target the nines of the adult, mainly the nines in this case, through the covers, so the natural or artificial covers, and the lethal, sublethal, and possible interference with the bacterium transmission when we target the adults through the olive tree. Regarding the direct and indirect effect, when we target, or in this case, our objective just targeting the nines, if possible, and the adults, to reduce the population and to try to reduce the number of specimens vectoring the bacteria, we have different results. Probably the first results were obtained in Italy by the team of Professor Bosco and Paola Dolci, that they perform an evaluation of different natural strains of entomopathogenic fungi against nymphal and adult stage of Philenos spumarius with very good results. But if we consider the third key point regarding the entomopathogenic fungi, that is the natural presence and diversity in different natural and artificial agroforestry systems, either in the soil, in the insect, or in the plants, our aim, the aim of the Cordoba team in collaboration with the CSIC team, is to try to look for active entomopathogenic fungi strain coming from the olive crop agroecosystem, coming from the olive tree phylloplane, or the leaves of the olive tree, from the soil of the olive crops, and from the leaves or usually natural covers in the olive tree, in the olive crop system. And we have evaluated different to now 15 strains coming from the olive crop ecosystem, and we have a very virulent strain. You can see in the screen the fungal outgrowth on a Philenos spumarius adult of one strain coming from the phylloplane of olive crop plant. That is probably to now the more virulent one that is being selected for different experiments, apart from strain 0158, that is a strain also coming from a soil that is also a quite important strain regarding the effectivity against this homopteran. And we have activity not only against adults, but also against nymphs. And these have been recently published, a manuscript reporting a very, very interesting efficacy of this strain in fungal spray against the nymphs and against adults via nymphs. We have reported in an experimental field close to the University of Córdoba, to the campus of Rabanales, with a standardized field experiment in a random design. We had found a good Henderson-Altington efficiency. Usually this is a typical formula to calculate the efficiency of the entomopathogenic fungus against the nymph population. You can see very nice effectivity. And the pebbling formula to calculate the relative reduction of the nymph. There is something happening. There is another speaker. So we have a very nice efficacy against the nymph population, in this case against the Neophilenus campestris population, but also quite important against the adult population via nymphs, with also an important variation in the relative adult population and a very nice efficiency. So using the entomopathogenic fungi directly spraying the cover, either natural or artificial, in particularly selected areas in which we have the risk, or they are particularly marked for the risk of transmission, or with high population of the vector, we could proceed by using entomopathogenic fungi targeting the homopteran community in the covers. But also, we can have an activity, remember, not directly targeting in the moment, in the particular moment in springtime. For instance, this experiment toward the nines had been performed in Cordoba in April, early April. We could also try to target the nine for the adult population through the plant. As I said before, trying to promote the endophytic colonization of the cover, either by fungal spray or by soil treatment, to finally have an effect on the Homopteran, the ninth Homopteran population. And we have demonstrated using different experimental designs, for instance, using Sonchus oleraceo, we have clearly demonstrated that when the adults, Philenus spumarius, feed, remember, we are not treating, in this case, the adult. We are not targeting the adult with the fungal suspension. Instead, we are colonizing the plant, and when the adult feed on the colonized plant, we had a very interesting activity through the plant. This is a key point, because we can have an activity towards the nines and the adults when they feed on colonized plant. We have used also typical components of the cover, natural covers or artificial cover in southern Spain, like Lolium rigidum. And we have demonstrated also that, first of all, most of the cover can be colonized by entomopathogenic fungi, either using soil or even seed dressing, soil treatments, or spray treatments. And when different, here you see Neophylenus campentris and Eoscius variegatus, when different possible vectors feed on the colonized plant, they become infected, and we can have a very important indirect activity through the plant. This is another quite interesting activity. Moreover, we have demonstrated that when we apply, we have developed in our previous work a strategy for controlling the olive fly by targeting the preimaginals in the soil in autumn, by soil treatment beneath the tree canopy, targeting the preimaginal of the olive fly. This treatment, either performed in autumn or in spring, are quite interesting also in order to reduce the Ochenurhyncha populations in the covers in different olive crops. In wide area experiment using farms of 50 hectares. And now, presently, we are trying to perform similar experiment, but not using the covers, but instead using the olive crop. This is an ongoing work, but I have to show you that using the typical plant that are used to establish plantations, we have demonstrated a quite interesting point. We have demonstrated that by applying the fungus to the soil and by spraying and by a standardized protocol to demonstrate whether the fungus may colonize the plant, we have demonstrated in 3 varieties, Arbequina, Picual, and Frantoio, that, first of all, the soil treatments may lead to always root colonization. I mean, we have demonstrated before that several of our studies are research competent, and if you see here, all the soil in the soil treatments, either 48 hours, 7 days, or 15 days after treatment, we only have colonization of the roots. On the contrary, when we perform foliar treatments, here you see in the 3 varieties, we may have, this is quite interesting, sometimes by performing a spray, we may have colonization of the leaves, colonization of the stem, and colonization of the root. It looks like the fungus is able to colonize and to move in the plant, even reaching the roots. But to now, we have not performed experiment to see whether adults feeding on these plants that have been colonized are killed or not. This is why this is an ongoing experiment. And even if we apply directly the fungal suspension, targeting directly the knife of the adults, or whether we do it by or through the plant. A key point also, as I have said before, is just not only to reduce the population of the vector population, but also to try to interfere with the transmission of the Xylella fastidiosa. And in this case, we have performed several experiments in collaboration with the team of Professor Ferreres and Dr. Moreno. And even we have published a first manuscript regarding or reporting not only the direct effects of the entomopathogenic fungi that I have shown you before on the lethal and sublethal, but also we have demonstrated, in this case, using again Sonchus, we have demonstrated, first of all, a high colonization of the plant. We have demonstrated an important mortality of adult feeding even on non-distant treated lead. I mean that we have demonstrated that in the Sonchus plant, the fungus have a systemic colonization of the plant. And interestingly, we have here different results in herbaceous plants like Sonchus and in the olive tree, because regarding the electropenetration technique, because we have demonstrated that the insect feeding on colonized plants reach the xylem vessel faster than insect feeding on non-colonized plant in Sonchus, but in the olive tree, we have found the contrary. That means that the insect feeding on colonized olive plants reach the xylem vessel later. It seems that they don't like really to feed on olive colonized plant. And this is quite important because we don't know what could be the effect of such observation on the Xylella fastidiosa transmission. Even in the frame of a fellow of Dr. Natalia González Mas in the team of Dr. Don Giovanni, we have performed a series of ISAs to unravel whether in this case using periwinkle plants, whether the endophytic colonization of the plant may lead to a reduction or not of the transmission of the bacterium. And this is also an ongoing work because we have different results and our results need a new repetition because as you may see regarding the Boveria bassiana strain on the left, not to the meta-region one, we have a qualitative reduction reaching significance at 10%, but not at 5%. I mean, we have the modification of the feeding of the insect on the plant is producing a reduction, but depending on the repetition, we have results that not allow having a significance in this point. This is also an ongoing work. And finally, our question is also how the feeding of the insect is or not altered when we have a colonization of a priming of the olive crop plant with entomopathogenic fungi. And in this point, this is a clearly ongoing work. We are performing this experiment in the last campaign. We are colonizing plant and we are performing for the first time, accordingly to Professor Ferreira, I think that this is the first time that they perform EPGs using the olive crop plant. That is a technology that had been not previously evaluated because the difficulties of using olive plant for such a technology. But our results are very important, and we hope in the very, very near future to have our first reporting regarding the behavior of the insect. I have said you before, we have a change in they reach the xylem vessel. There is a delay in the reaching of the vessel, of the xylem vessel by the insects feeding on colonized plant if compared with the insect feeding in control plants. And finally, even the Blanca has said at the beginning, we have introduced myself, has said that we have been all our lives in parallel research lines, but our lines have crossed many times with very, very interesting results. And in this project, we are performing a parallel line using entomopathogenic fungi in which we are we are developing experiment to try to look for bactericidal compounds secreted by entomopathogenic fungi. The same strain I have shown you before, we are developing standardized protocols in my lab, the fermentation, and we are obtaining crude extracts, and even we have performed several purification steps for bactericidal activity against Xylella fastidiosa. And we have 2 crude extracts. Here you can see the effect at a very important concentration, 50% concentration of the fungal extract. But we have selected, and this is also ongoing work, the crude extract for 2 strains that had a very important reduction of the Xylella fastidiosa growth, only at 2% concentration of the crude extract. We would like to perform purification step and to see whether we have any interesting molecule at this point. And well, I am available for you. I think it's time, if you have any questions, to try to guarantee the future of the olive crop. The present is good. The past was also, we have to guarantee the future. Many thanks again, Blanca, Anna, for making me possible to participate in these Xylella seminars, and I am available for any question.
Thank you, Enrique. So, as you know, you can either make questions in the chat, typing what you want to ask Enrique, or just raise your hand and you can speak in the microphone.